Tonight we're joined by Dr. Jessica Petersen from University of Nebraska. Most of you know Jessica very well, but she's going to visit with us tonight a little bit about thinking about how to develop maybe a genetic test for unwanted traits and share some information along those lines. I think there's a lot of discussion in the last 6 months to a year around kind of this topic. And so Jessica, why don't you go right ahead and share your screen and you can take it from here.
Okay, we'll do it. All right, so look okay?
Looks good.
Okay, thanks. So it's good to see you all. I'm going to go through how we develop genetic tests for unwanted traits, and like Jeff said, I get asked a lot is how do we get a test for something? And so I'm going to go through what I decided were 7 steps to identifying genetic variants and getting the result and getting that genetic test available. And I'm going to use it. I'm going to do this using an example from a previous project that Renee Seek, now Renee Schroeder, was a graduate student leading in my lab. That's actually a defect in Herefords. But the fact that it's in Herefords doesn't matter. The process would be the same if it were something in yak. So I start with this. It happens. Mutations happen. And in humans, there's about 30 trillion cells, and almost all of those cells have DNA in them. Any one day, we replace over 300 billion of our cells. I'm guessing that number is probably even larger in yaks just because they have a bigger body area. So every time that a cell has to replace itself, they have to divide, which means they have to replicate their DNA. So when they replicate the DNA, it's like copying this really, really long code. It's 2.5 billion little letters of DNA, and mistakes happen. So it doesn't always copy correctly. And in fact, there's estimates that every time DNA replicates, there's from 1 to 10 new mutations that's in that new cell. So those probably don't matter because it's not in the DNA in your whole body, it's just in that single cell. They do matter if that mutation could cause cancer or something like that. You could get localized issues. But generally, those mutations don't affect our herds, for example, if we're thinking about our yak. So if they are fixed, those mutations are present in the new cell. The mutations then that we are interested in, ones that could affect the health of our animals, are mutations generally that arise early in the embryo, because then they're going to be in a lot of cells in that embryo and then in that adult yak, or mutations that originate in germ cells, so in the sperm or the oocytes of the yak, because if a mutation happens there, then it is also going to be present in the whole yak once it's born. And then those can be passed on to the next generation. So here are my 7 steps to identifying causative genetic variation. We're just going to go through each of these again with my example of mandibulofacial dysostosis in Hereford cattle. So step 1 is clarify the phenotype. And this picture is up here because all 4 of these calves have hydrocephalus. So hydrocephalus being fluid on the brain. And if I were to get a report of a yak with hydrocephalus without more information than that, and then another report from somebody else, I potentially could be classifying all these calves as having the exact same phenotype. So it's really important to be as specific as possible when we're identifying the phenotype. So if we actually went in and opened up the head of that first calf, this is what you'd see. There's really no neural tissue at all. This is an example of Angus neuropathic hydrocephalus. This was the calf in the top right corner. I believe, I'm not 100% sure, this was Dr. David Steffen's slide. I believe that it had a viral induced case of hydrocephalus. Here you can see the brain stem is fairly straight, and this is the cerebellum of that calf's brain. And then this calf is one that is in a study that I've been involved in. And this is another case of hydrocephalus. In this case, the cerebellum is shrunk and that brainstem is twisted. So it's not likely, and I'm certain that all 3 of these cases, and 4, if we go back to the previous slide, of hydrocephalus were not due to the same cause. So to find a genetic cause, we need to be really, really careful in that we're classifying all of our phenotypes correctly so we can focus on a single condition or one condition at a time. So when something gets reported, or to Dr. Stefan, who's a veterinarian that I work with often, or to myself, there's a few online resources that are useful, and these are ones that you could use yourself if you see a condition pop up. One is OMIA, so it's Online Mendelian Inheritance in Animals, and you can just put a search term in here like hydrocephalus, and it's going to give you something like this. The search here was hydrocephalus cattle. It's going to give you all the instances that have been reported of genetic hydrocephalus. Sometimes it will tell you the gene if it's been identified, or it will tell you more information about those cases. So it could be that something that you're seeing is something that's already been reported. So this is one of the first places we'd look to either get an idea if it's been reported, or to get an idea if a gene has been identified, what that gene is and how that might be causative of a condition that we're seeing. So clarifying the phenotype is, like I said, extremely important. And I think at this point, it's often valuable to get a veterinarian involved. I do genetics. I'm not a veterinarian, but veterinarians can rule out environmental causes, so toxins or viral exposures or mineral deficiencies, things like that. To do that, it's often good to have blood samples. So if the calf is alive when it has a apparent defect, if you can get blood, both red and purple top tubes. A sample of the liver is very good for testing for toxins. And then depending upon what the phenotype is, a relevant sample of that part of the calf's body is helpful. So if we have dwarfism, Dr. Stefan likes to see limb bones. If we have cleft palate, getting the head, or hydrocephalus, obviously having the head is helpful. And he'll often get whole calves. So people will throw them in the freezer and then ship them to Dr. Stefan. So that generally doesn't screw up his ability to look at the calves and diagnose what might be going on. And also having the calf itself frozen, or any piece of tissue, is helpful for me because I can get DNA from that to investigate in the future. So if we rule out environmental causes, or if we're still not quite sure, we might look to genetics when that condition is found in more than one herd, because that it's unlikely that in multiple herds there's the exact same management conditions or the same toxin exposures and that sort of thing. Genetics is more likely if there's other calves in the same herd that are not affected. So again, if it was a toxin that all the cows were eating, you would expect all the calves to have some sort of deficiency. And it's also more likely if we see multiple cases over time. And so I mentioned contacting a veterinarian or researcher. This abnormality report came online earlier this year. The link is there, and we could add this, Jeff, I think, to the recording of this presentation just to make sure everybody has it. But it's really important that if you have something that you might think is a one-off, that it gets reported, just because maybe somebody else saw the same thing. And if that's the case, it could be something that might be genetic that we might want to investigate. So on this Google document, it just asks you general information about the calf's ID, its sex, its parents. It has you describe the condition, and general information like that. It also tells you, if possible, to take a genetic sample. The calf is dead, just cutting off a piece of the ear is really easy. If it's alive, blood is my favorite, but any sort of tissue sample of the calf, of the sire, and of the dam. So this report comes to me. It doesn't go to the yak associations at all. And just as general practice, anything that's reported for any of these cases in my lab, the animal IDs and all that remains confidential. So I know that generally everybody's interested in helping everybody out, but I also know that there can be some hesitation to report things. So I don't want that to be the case. None of this will be communicated with any farm names or any animal IDs. Okay, so I'm going to jump back to my Hereford example here. This was the spring that the pandemic started. The Breed Association contacted myself and Dr. Steffen and said there was a producer who had a calf that looked like this. And they reported it because last year they had a calf that looked like that. And the producers noted that the prior year they thought, oh, bad luck, something weird happened. But when it happened again, they noted that, oh, these dams are fairly related. And maybe this is something we should be worrying about. So unlike our hydrocephalus example, this phenotype is really distinct. So we weren't worried that if we had a Hereford with a curved jaw, and this was kind of the hallmark of this, they have these skin tags often at the end of their jaw and sometimes by their ears, that we had the same condition. So this was really fascinating because when Dr. Steffen dissected out this calf, so this picture here, this is that skin tag that you see on the jaw of that calf. It's connected to this cylindrical piece of cartilage that turns out to be what's called Meckel's cartilage, and it's encased in bone. So here's bone that sits around it. This cartilage is supposed to go away in utero. So it's there early in development, and then it gets ossified into the jawbone. So for some reason, these calves, that wasn't happening, and it resulted in this malformation of the jaw, the lower jaw generally. Some of the calves had a cleft palate, and they had weak master muscles. So even if they were able to get up and try to suck, their jaw muscles weren't very good to help them with that. So we had our phenotype pretty distinct. So something obviously not normal. It was very similar across calves, although the severity of it differed a little bit. So step 2, determine the pedigree. First part of that is to confirm parentage. And I've actually had people ask me questions about something that they think is interesting about their animal. And I say, well, have we confirmed who the sire is? And they're like, oh, no, but we're sure. And then we do that using DNA, and it's not actually that sire. So that's really important to do. If you cut off a piece of the ear and have a sample of that sire and dam, we can do that quite easily. And then we start to look at relationships among the affected calves. And so in this picture here, these are the first 6 reported calves. And you can see that these are all their parents, and all their parents within 3 or 4 generations go back to the same bull. So in that case, we were thinking this is probably a recessive mutation that all these calves were homozygous for that led to this condition. If this wasn't this clear, we'd be asking ourselves, is there one bull that was the sire of all these calves? That could denote a dominant trait, and I'll give you an example of a dominant trait at the end here. So we look for patterns. We don't have to have a perfect idea of how we expect this to be inherited to do the genetics, but it definitely helps us. We have an idea of what pattern we should expect in the DNA. Okay, clarified the phenotype, determine the pedigree. Number 3, get those DNA samples. Of the affected animals, all affected animals that you can find, their parents, any possible founder or any links between those parents and who we think might be connecting them together. And then probably almost as important as the affected animals is having information or DNA from related normal animals. Especially in yak, where we know they're all fairly closely related, having DNA to rule out what variation is present just within that family, and parse out that from what is only in the affected animals is really important. So the DNA samples can be anything. I've had a lot of people go to the dead pile and dig up a half-rotten carcass to get me a piece of muscle or for DNA, and that works. It's better if it's fresh and it's less smelly to ship, but any sort of piece of DNA, this is liver, skeletal muscle, pulled hair, we just need the roots, and EDTA blood, the purple top blood is really good. So you have DNA, we have an idea that we think is genetic, and then we're going to go ahead and sequence that DNA. There's 2 approaches we could take. One is what we call a candidate gene approach. And so if we go to that website I mentioned before, or maybe to PubMed, where a lot of human literature is published, we could search for things like abnormal cartilage in the jaw or something like that. And we could see if there's any similar conditions in humans or animals. and if there's known genes that are associated with that. Okay. So in our case, we were pretty sure that something was wrong here. So during development, this Meckel's cartilage, that tube that I showed you, forms, and then later in development, it gets ossified or made into bone, and it gets made into that mandible or that jaw. When we started this, Dr. Steffen said, I bet it's going to be one of these genes, DLX5 or 6, because that's important in this step here to get that process to happen. But I wasn't confident enough that we could rely on it going to be one of those genes. And so we took the approach of whole genome sequencing. So instead of sequencing just one gene or a couple genes that we think might be important, our whole genome sequencing gets the whole entire genome. The advantage to this is that if we were wrong in guessing that candidate gene, we would still have sequence of the whole entire genome, the rest of it that we can look at. So we don't have to go back and start over with a new candidate gene. Whole genome sequencing is actually quite inexpensive these days. I've been paying around $500 an animal, and I just got a quote for a different project that was closer to $300. So we can sequence animals quite inexpensively, and that lets us look at all 2.6 billion bases of the DNA. So we sequence it, and then we're going to evaluate the data. This is the search for the needle in the haystack. We're searching through that whole entire genome. And in this case, where we think it's a recessive, we're asking the question, what do the affected calves have that the others do not? And specifically, where are the calves homozygous or have two copies of the same DNA base? All their parents then needed to be heterozygous. So they needed to have one of those bad copies, if you will, to give to their calves. They can't have two bad copies, though, because otherwise they would be affected themselves. And then we have relative animals, and we can't be for certain if those animals are carriers or not. So we expect that those related animals either don't have the variants or they're heterozygous for it. And then, especially in this case of Herefords, where we had it pretty much restricted to one family, we also assume this would be absent in unrelated cattle. So not only unrelated bloodlines of Herefords, but absent in Angus and Brahman and Charolais and all of those. So here's what the process that Renee went through when she analyzed these data. We sequenced 3 affected calves, 10 parents, so parents of these calves, and then parents of calves that had been disposed of or previously born, that they didn't get samples of those calves, and then 7 related individuals. And these included sibs of the cows that were parents and a couple sibs of the affected calves. So amazingly, Renee was able to filter the 2.5 billion bases of the genome down to 143 variants that fit our hypothesis. So where the cows were homozygous, their parents were heterozygous, and then the unrelated animals didn't have that variant at all. So we accessed other whole genome sequence data. Some of it was Herefords that the association had sequenced for other things. Some of it was the 1,000 bulls database of sequence. We were able to rule out of these 143 all but 8. So things that we ruled out included things that were homozygous in other animals, because these other animals weren't reported to have this disfigurement. If we wouldn't have, those people wouldn't have sequenced them and deposited their sequence in these databases if they weren't normal. So we're able to get that down to 8 variants. We went to the public archive. We were able to eliminate 2 more by finding 2 of these variants that were homozygous in multiple other animals. And then we used Sanger sequencing because we got DNA on another affected calf from more parents, our suspect founder and his sire, and other related animals. We were able to narrow these 6 variants down to 2. Those 2 variants are shown here. The first one is a missense variant, so it changes an amino acid in the protein of this gene, CYP26C1. The other one was just a variant that's in the middle of genes, and it wasn't predicted to change anything in the genome. So at this point, we focused in on this variant as potentially the causative for this condition. And again, we got here because we're filtering through all the information we had to find things that only fit that pattern where those affected calves were homozygous. So Hereford then sent us a whole bunch of DNA on other animals. So I have them classified here based upon the relationship to our presumed founder, that bull that was in the top of that pedigree that all of our parents tied to. So if the animal that we sequenced had that bull in either the maternal or paternal pedigree, so just one side, we found that 2/3 of those animals were normal. So this is the TT normal genotype. About 1/3 of them were heterozygous, so were carriers of this mutation, the C that we are tracking, and none of them were homozygous. Okay. So then if we looked at the animals that had that founder bull in both sides of their pedigree, so both mom and dad were related to him, we found 50 some animals that were normal, about 50 that were carriers, and these were our 5 affected calves that we had DNA on. So the only ones that were homozygous for the C were our affected calves. If the Hereford is not related to our founder at all, they're totally normal. None of those animals had the C allele that we think is causative, and that founder himself actually was heterozygous. So that makes sense. So we're able to genotype about 800 animals. We showed that this mutation, this C, still fit our pattern. And so we're pretty confident at this point that we found something that's causative of this facial malformation. So once we found it, now step 6 is sometimes really hard. We need to validate this, which means we need to gather a bunch of evidence to show that this mutation is likely to be causing this condition. So I asked myself a few questions. One is this mutation in a gene that makes sense? So based upon what we know about that gene, could it lead to that facial malformation? Is that mutation expected to alter gene function? I already told you that mutation in this gene is supposed to change the amino acid, and I'll show you a little bit about that in a minute. And is that part of the genome, and that gene in particular, expressed in the relevant tissue? So there's 20-some thousand genes in a yak genome, and they're all doing different things. They're all on and off at different times and in different tissues. So genes that are important to the liver might not be expressed at all in the skin or in the eye. So we also need to show that that gene is expressed in the relevant tissue that could lead to what we're seeing. Okay. So we worked with a biochemist who's in the veterinary school who did protein modeling. And so this gray structure is a model of this protein that this gene makes. And this is where the gene that our mutation is in. And so it has some pretty cool alpha helices, some coils. And what's shown in this red box is this site where this protein interacts with retinoic acid. So this red is retinoic acid that goes and interacts with that, or that the green is a retinoic acid that interacts with this protein. And so this part of the protein is really important. So our mutation is actually here. It's purple. If we zoom in, we see it over here. And at first I'd be like, well, I don't know if that's going to interfere with the job of this protein, which is to interact with this stuff over here. But doing more modeling, this is looking at that sequence of the amino acids. So basically the sequence of that protein. And this red bar says that this software predicts that this whole area is that alpha helix. So it's this spiral structure. When our mutation happens, it changes this L amino acid into a P. And our modeling says that's going to break this alpha helix structure. Okay. So this might not be how it works, but in my mind, I now picture this with our mutation. We have this helix, and maybe it bends here, right? Or does something that potentially interferes with how things can interact with it. So it's likely to alter the structure of the protein. So I also said it's good to see if that gene is expressed in places that make sense. And so we went to the literature, and we found that this gene is involved in retinoic acid degradation, okay? And it's expressed in the maxillary and mandibular process of the first pharyngeal arch. All that is a bunch of scientific term for, in this little tiny embryo, that gene is really important in this part of that embryo that turns into the jaw. And so this gene is actually expected to be expressed in our cattle embryos around 30 days of gestation. So really early is probably when this defect had this impact on development. So if you remember this picture, this is where Dr. Stefan guessed our defect was going to be. He wasn't quite right, but it turns out that this retinoic acid signaling is important right here to stimulate this step to get that process going. So if we would have guessed and done a candidate gene approach, we wouldn't have found it, but we were in the right pathway. So there's some other cool studies both in zebrafish and in mice that show if you knock down this gene, or if you take that protein out of that animal as it develops, they can't process retinoic acid appropriately, and that leads to abnormal development of those jaws, whether it's a zebrafish or a mouse. So again, this adds to our evidence that this gene is important to the feature that we're interested in these cattle. Similarly, that same thing happens in mice. We knock out those genes, we being scientists, not me, there's atypical development of those pharyngeal arches and the jaw bones. So more evidence. Here's what I'm excited about, where animal research can be important beyond the animal, is that there's a couple conditions in humans. This first is focal facial dermal dysplasia type 4, where these little abnormal, I don't know what to call them, almost like little scar formations, but this is along where the Meckel's cartilage would be in humans. Or this one's really striking to me, hemifacial microsomia in humans, that has this jaw malformation, has this little skin tag, which looks a heck of a lot like our Hereford, right? And these humans also have soft palate issues. And like I said, some of our calves had cleft palate, and the humans have weakened muscle development. So both of these conditions in humans are attributed to mutations in the same gene that we're investigating for Herefords. So again, it's more information that we do in fact have the right gene and the right mutation. And what I hope, because I have several of these projects now, we can't, it's hard to study development and treatment of these conditions in humans. Man, we could breed Herefords with it. And we could, those human researchers could study those embryos as they develop and potentially learn something about therapeutics for humans. So far, that hasn't gotten picked up to my knowledge, but I think it's an opportunity with animal models that could be used. Okay, so we've gotten through our 6th step of validation, gathering evidence that that is the right gene. And then step 7 is to communicate the results, not only to breeders, but for me, it's important that this gets communicated to the scientific public and peer reviewed, so that other scientists can read this and say, oh yeah, I think you got it. I agree with your conclusions. Or sometimes they say, no, I don't agree, and they send us back to the drawing board. But I'm really proud of this example because, like I said, it was reported to us right before the pandemic. We shipped samples to sequence the week before we were shut down. I was able to get my grad student in the lab while it was shut down to do a lot of this genotyping. And we had this published about 9 months later, and this test was available. So this was a case where just all fell into place, and we had really good cooperation from breeders and the Breed Association to get samples all together. And they announced the commercial test in August. And again, we started sequencing in March of that year. So this test is available at Neogen for breeders to use. So that was my example of a recessive. Those are easier than other mutations because we have an expectation that the parents should carry whatever it is, and our calves should be homozygous, and we can eliminate a lot of other variation fairly quickly. One other example that I think is a fun one that's not a recessive is this. So we just published this earlier this year. This is an Angus operation, and they went out to check their cows in the morning, and this little guy was here. So I think that's everybody's first reaction, whose Holstein got into the herd or something like that, right? But we parent verified, and both parents are purebred Black Angus. And so in this case, we did whole genome sequence. We clarified the phenotype, which was that calf is white and he shouldn't be. Determined the pedigree, you got DNA, whole genome sequence, mom, dad, and the calf. And here's what we found. So here's what our whole genome sequence looks like. The sequence of the DNA is on the bottom here. So those are the actual bases of DNA. Each of these gray bars is a sequence. So in the calf, you can see that there's these spaces here that have the number 3 in them. That's saying there's 3 bases of DNA that are deleted in that calf. That wasn't present in the sire. It wasn't present in the dam. So we did another type of sequencing to confirm it. And here's our dam, here's our sire, and then you see how things get kind of strange down here at the calf. It looks like there's multiple signals. So each of these humps is a different base of DNA. And we confirmed that that calf did indeed have a deletion that neither mom or dad had. So this is an instance where when DNA replicated, that calf had a mutation that was brand new to it. And so mom and dad are totally normal and this calf is not. This mutation happens to be in a gene that is well known to be involved in coat color. So this gene is called MITF. It causes our merle coloration in dogs. Often if a dog is homozygous for merle, if it has 2 copies, they might have small eyes, they might be deaf and have sight problems. We have no indication that our calf had any hearing or eye problems, but he was never evaluated other than the breeders trying to, wave and make noises and see if he responded. This MITF gene mutations also cause white patterning in dogs, splashed white in horses. And interestingly, there's a mutation, these are German fleck B calves, mutation like right next door to the one that we found in this Angus, causes these animals to be white. It's also a dominant mutation. They also have hearing loss and then ocular issues. So they're not seeing properly as well. So validation in this case is really hard because unless Casper, which is what they called him, was able to breed and we could show that when he passed this mutation on, his calves all had the same thing, I have no way to show that this mutation is for sure positive of this condition. All I can do is put together the information that this is a brand new mutation that he had and his parents didn't. And it disrupts this gene. And we know that this gene, when disrupted, causes white in other animals. So an example of a dominant mutation, a de novo dominant mutation in an animal. So all of this is only possible because breeders and breed associations helped connect the dots. They report these things. The breed association, in this case, the Hereford Association, was concerned enough, or they noted, wow, this is not the first calf that was like this from these producers. These animals are related. We might have an issue. And when they started looking for other breeders of similar bloodlines, they found other issues. So then they're able to get us those samples and put it all together. So this is Dr. Steffen. He is an encyclopedia of information on cattle defects. He's my first go-to if anybody calls me. I'm the geneticist. And says we might have an issue. I get his input, but we all work together on this, and this is my team of students right now. So with that, I'm happy to have a conversation, answer some questions, and have a discussion.
Dr. Petersen, thank you so much for sharing. It's been a really good session tonight. And if you've got any questions, be sure to send an email to Dr. Petersen or send it to us, and we'll be happy to pass it along. Also, I'll be working on getting the video edited down, and I'll put that up on YouTube, and we'll send that link out, and you can share it with other folks that couldn't be on this evening. And we'll make sure to get that abnormality link both for the pre-2020, 2023, I guess it was, and the new one out as well. Thank you all. Dr. Petersen, thank you so much for joining us. We really appreciate it.
Yeah, this is fun. Jessica. You guys make this a fun group to work with. So appreciate it.